Hello everyone. I hope you are all fine and it's going good, I think so. Today we are going to see about the anatomical aspects. I hope you know well about anatomy. What is anatomy again, right? So anatomy is nothing but the study about the internal structures of a plant system. So that is known as anatomy. And next thing we see means anatomy. What are the things we can find out? Not only the internal structures. Okay. What are the things are being found in the external morphology also we can study, right? So anatomy is nothing but about the study of internal organization of a living organism. And the histology is nothing but the study about the tissue system. So you know well about cell. What is cell? No? It is a structural function of a living component. So each and every cell joined together to form a tissue system, right? And all the tissue systems will be grouped together to form a organ system. So not only in plants, in animals also, these things, not only in animals, in human beings also, these things have been happening. So it is a common phenomenon, plants, animals as well as human beings. But some physiological aspects will be varying. So how does, coming to the plant, so what are the components are being found in a plant now? Basically we can divide the plants into two systems. One is a root system and another one is shoot system. But anyhow, there is something else that makes the plant to grow. Right? So ஏதோ ஒன்று இருக்கு அதுதான் plant வந்து grow பண்ண வைக்குது. So that what you see means that is the growing point is being found in two places, right? Where are they? Upper system and as well as the lower system. That is, we are telling it as a shoot system and a root system. So in the shoot system, at the apical point we can find the growing region, and below the root tip we can find the growing region. So these 2 points makes the plant to move on from primary stage to secondary stage to become into a big tree again. Okay. So these things which are making the plants to grow up are known as meristematic tissues. That is a tissue that is known as meristematic tissue. Basically, this tissue can be divided into two categories. One is meristematic and another one is permanent tissue. So what is meristematic? Meristematic is nothing but those cells will be always in a active stage of division. That is known as meristematic tissues, right? So does this division happens continuously? No, right? So at particular stage it stops. So then only we can differentiate the system-wise arrangement. So namakke pathina we are growing system, we are having childhood, adult, then older stage. So the growth will be moving on into 3 stages. So likewise in plants also it happens so. So meristematic அப்படின்றது is the youngest cell. Okay. So there your differentiation will be moving on. So the cell once starts to divide now, or a particular stage the division stops. So that thing we are doing as a differentiation. Okay. So this permanent tissue again starts to divide, and that gives rise to simple tissue and complex tissue, right? So simple tissues we see means they are nothing but, you know well, you are taking the section, you are observing the practical aspects. What are the cells you are able to find out? Parenchyma, sclerenchyma, all these things we are able to find. The next category is complex tissue. So there are main two things are there. One is water conducting tissue, that is food conducting tissue, xylem and phloem. That's it. So shall move on to the next thing. What are the characteristic features of meristematic tissues? And before that, we can find a finalized chart of whole along the tissue components. What are they? The tissues mainly categorized into meristem and permanent. Again, that permanent is being classified into simple and complex. Okay. And in meristematic tissues, where are they are being originated, right? So அதோட ஃபங்க்ஷனை பேஸ் பண்ணி எங்க ஆரிஜின் ஆகுது அப்படின்னு பார்க்கும்போது we can classify the meristematic tissues also, right? So we can classify that based on origin, primary, secondary அப்படின்னு we can categorize. In the position it is being placed அப்படின்னு பார்த்தோன்னா it has been divided into apical meristem, intercalary meristem, lateral meristem. So apical meristem will be giving rise to the length of the plant system. Okay. Then the lateral meristem will be giving the girthness of a plant system. So likewise we are having many systems of meristems. But today what we are going to see is basically about the secretory tissue, right? So permanent tissues. In permanent tissue, the third category is what? Special kind of tissues, right? So in that, what are the thing you are able to find out is internal secretory tissues and external. Internal means inside, external means outside. You can find out. So we can see in detail in the next. So what are the categories? What are the characterization of meristem? Right? I told you, no, they will be in a continuous state of division. அப்படியே செல் டிவைட் ஆகிட்டு போயிட்டே இருக்கும். அதோட வேலையை மட்டும் என்ன குட்டி போட்டுக்கிட்டே இருக்கிறது. That's all. So that's the thing will be happening. And அந்த meristematic tissue was first coined. யார் இந்த term coin out பண்ணாங்க அப்படின்னு பார்த்தோம்னா, it is nothing but the person who is known as Karl Wilhelm von Nageli. So we used to tell Nageli. We used to tell him. Okay. So next thing is shoot system, root system, right? So the shoot apex you can find the growing point. The root apex you can find the growing point. So this is meristematic tissue, right? So the cells are nothing but they will be isodiametric in shape, right? And they will be having a prominent nucleus will be found, and dense cytoplasmic content can also be seen in that meristematic tissue. Okay, and vacuoles cannot be seen there. So that is the basic characteristic features of a meristematic tissues. These are some cells, as you told. That is parenchyma, collenchyma. You have seen in the section. Also, so those are the cells that we can tell it as a simple tissues. So parenchyma, collenchyma. So the role of parenchyma is they will be acting as a storage agent. Collenchyma means it is mechanical end support, right? And sclerenchyma is also mechanical end support. The basic difference what you can say is parenchyma, collenchyma are living cells, right? But sclerenchyma, dead cells. But in the early stage, they will be in a living stage only. Later on, at maturity only, they will be moving on to a dead cells. Okay. Next, these are the things that I told you about permanent tissues. So permanent tissue, they will be having some role functioning. That happens after the differentiation stage. Right? So today, you are going to see mainly about the secretory. Have you observed secretory tissues? Have you seen any secretory products in your plants? Other things. What are they? Latex. Very good. Then resins, tannins. Okay. But actually, regularly you used to find nectaries, latex. Okay. So whether what kind of thing we can find in this plant? Latex, right? So almost this secretory structures have been working on. Most of the plant have these structures, right? Especially in U4BAC member, in Asclepiadaceae member. So this secretory tissues order in a role by finding out these secretory structures, we can act as a taxonomic aid. So in the family or latex, but this plant family belongs. அப்போ நெக்டார்ஸ் நிறைய இருக்குன்னா அந்த மாதிரி பிளான்ட் வந்து ஒரு ஃபேமிலிக்கு பிலாங்ஸ் டு அப்படின்றத நம்ம so we can use this secretory tissues to find out the taxonomic age in a family it belongs to அப்படின்றத நம்ம find out பண்ணலாம். Okay. For example, in medium. Before that I'll tell you. Next slide. In secretory tissues, what are the things you are going to see means first what is secretory tissues, right? And next what is secretory products and what are its economic uses. There are economic uses we can find, and any ecological adaptations are being found we can find out. So one by one we can move on. So what is the characterization? So as I told you, it is found almost in all the parts of a plant system. It may be found in a external வெளியில பார்க்க முடியும், இல்லனா ஒரு செக்ஷன் எடுத்து internal aspect நம்மளால பார்க்க முடியும். Okay, so these cells are known as specialized group of cells. Not all the cells will be secreting the secretory products. Particular cell alone take charge. So how we are taking charge for a particular function or a representative and some other characters we are giving you know. So likewise, these plants also have some special structures. That structures alone secrete the secretory products, right? Those cells that secrete the secretory products are known as secretory cells. That's all, very simple, right? So there are many things. One is Sasapoidida, பெருங்காயம் நம்ம சாப்பிடுறோம் இல்லையா? So it is a secretory product. நிறைய பிளான்ஸ் we used to see முருங்கைக்காய் சாப்பிட்ருக்கோம் இல்லையா, நம்ம முருங்கை மரம் பார்த்துருக்கோமா, பார்த்துருக்கீங்க. அதில் என்ன வரும் ஜாஸ்தியா, மரத்தோட வெளியில பார்த்தீங்கன்னா ஒரு மாதிரி கம், that is also a secretory product. But in your childhood we doesn't know what it is, so something is coming out அப்படின்லாம் நம்ம நினைப்போம், but it is a secretory products, right? அதில் then neemல in all these trees we can find the secretory products. What are the economic importance? So does these products have some economic importance? Yes, right, very good. So they have some economic, for example rubber, latex. Okay, so in area, the milk that is being coming out from a rubber plant, it is economically useful, right? Then pulp is being used, likewise resin, lubrication, vehicles for lubrication எடுக்கிற oils எல்லாமே வந்து they are also being taken from a, so they are also a, and by having these secretory products, some plants, these animals doesn't go and eat the plant. ஒரு பிளான்ட்ல லாட்டெக்ஸோ மில்கோ ஜாஸ்தியா இருக்கும், அந்த பிளான்ட்க்கு these animal doesn't go and raise, so it is a protection to a plant. Okay, so these are some ecological adaptations that we can find. These secretory tissues can be classified mainly into two forms. So they be categorized, எதுவா இருந்தாலும் நம்ம categorize பண்ணுவோம் இல்லையா, one is external and another one is internal. So coming to this external part, we can find these structures, அதாவது nectaries, வெளியில நம்ம பார்க்கணும். Okay, by observing ourselves we can find out. So nectaries, then hydatodes, all trichomes, hair like structures நம்ம பார்க்கணும். Then அது மட்டும் இல்லாம Dracena, insectivorous plant, you might have seen earlier. So அந்த மாதிரி plants we can find the external structures and all. Okay, so for best example we can find this. This is Calotropis, you know well. Okay, so once if we peel here or if we harm this, we can find the latex will be coming out. So this latex is having a, is there any use of it? We are having medicinal uses we are having. So likewise, this is, you can find nectars, நிறைய honey எல்லாமே இருக்கு. So honey secrete பண்ணக்கூடிய ஒரு thing, structures தான் we are doing as a nectaries. So butterflies will be coming out, they will be, with the process what happens, pollination will be moving on. So that is a kind of nectaries we can find. So in this Euphorbiaceae nerium plant also we can find latex. Okay, whitey latex இல்லாம ஒரு transparency latex நம்ம இதுல பார்க்கணும். But Calotropis la பார்த்தோம்னா white colorல latex observe பண்ணணும். So here also we are having nectaries in this. So honey will be secreted. So these are some special things that we can find in the plant system. Yes, these external secretory structures. So you can find this. So what we are able to find? The picture tells something. Yeah, yeah. So resin is being coming out from your plant, and latex is also being coming out from the plant system. So external secretory structures, that is finding out, can categorize into hydatodes, nectaries. So nectaries I have told. Hydatodes is a thing that we can observe early morning, right? Funny season la pathona. We can find in the plants some dewdrops can be seen. So actually it is not a dewdrops, right? It is a process where the guttation process will be going on. So அங்க என்ன இருக்கு அப்படினு பார்த்தோம்னா stomata like structure, that is known as hydatodes. Due to water pressure, the water will be coming out during the night time. So in the early morning if you go and see means we can find the droplets near the laminar portions. Okay, so அதுதான் நம்ம hydatodes அப்படின்றது we will be telling you. So next பார்த்தோம்னா trichomes. So these trichomes we can find in which plants? Insectivorous plant, right? Dracena, Nepenthes. In these plants we can find the trichomes. Trichomes are nothing but they are hair-like structures, right? So on the hair-like structures we can find some attractive colors and some fluids will be present over the nectars. So once the insects, by having the attraction over the colors and nectars, they will be coming and sitting over the things. So once if it sits now, what happens? You can find the trichome. It closes the insect. So it gets captured. So later on that insect will be digested. The nitrogen which is needed for the plant will be taken by itself. So these nitrogen deficiency plants will be having this trichome-like structures. Okay. Next is nectaries. So nectaries is nothing but honey. Okay, sweety, a sugary fluid will be secreted from that particular gland. So you might have seen it, I have shown you also now. So, and that itself we are having floral nectaries and extra floral nectaries. We are having two types. So as I told you, hydrothods you can find. Hydrothods in the leaf margins you can find the dewdrops. Right? It is actually not a dewdrop. It is a process where the water pressure, the water will be coming out. It happens during the night time. So that process is known as fertilization. So you can find the internal physiological changes that is being going on. So there is a physiological changes where that cell can also be known as water stomata. Okay, actually stomata function is transportation, exchange of gases. But here the same role will be played by this hydrotherm. Okay. Next one if you see means internal secretory structures, right? So in internal secretory structures, now just by taking the sections, we can find out what are the things we are able to find. Okay. So in this internal secretory structures, we are having different types. One is secretory cells, then secretory cavities, in our ducts are lung. The last one will be the latticeous. Okay. So in internal secretory cells, there are many for example, we used to take papaya, no? Papaya, yarn. அதெல்லாம் சாப்பிடும்போது நமக்கு என்ன ஃபீல் ஆகும் சிலருக்கு இல்லை அதை க்ளீன் பண்ணும்போது any irritations that you can find itching will be there. So the itching is being caused by some structures, that is internal cells. They are known as raphites. Okay, that is nothing but a crystal-like cells. So in internal cells, secretory cells, we can group the raphites. Okay, so these raphites can be found in papaya, yam, அது நம்ம சாப்பிடும்போது என்ன ஆகுனா just it injects into our body. So that's why it gives itches to us. Okay, so that is nothing but the secretory cells will be there. So that's why when we used to cook, we used to put some salt and make it to remove all these crystals. So next is latticeous tissue, right? So latticeous tissue, if you see, means it is nothing but those things that secrete a latex. So white fluid I told you, milky-like thing, that is known as latticeous. Almost this latex is found in many plants, especially you can find in 12,500 species we can find this latex. Okay, and more than 900 genera we can find out this latticeous tissue. For example, Regnilidium plant we can find in Havia, rubber sonoria. So in these plants we can find out the structures. And what are the general characters of it? And what are the roles of it? These are some roles. So that is some parenchyma cells. So not some special structures. Some parenchyma cells will be modified by itself to secrete all these products. Okay. So this latex may be white in color or it may be yellow in color. So depending upon the plant system. So you can find in these plants. Example, in white color you can find in Fabaceae and Cannabinaceae. Colorless we can find in Moraceae memberla we can find it. Okay. Then Apocynaceae member. So in Moraceae, what example you can tell? Very good. So இது எல்லாமே நம்ம find out பண்ணலாம் latticeous tissues. Okay. So next, these latticeous tissue we can divide into two categories, right? One is articulated latticeous and another one is non-articulated latticeous. So every time the cells, how the cells are being formed, based on that we are telling it two types, right? So coming to the articulated latticeous, it is formed of a single component. So articulated latticeous are formed of a compound in origin, right? So many cells will be grouped together to form an articulated lattice. So it is not formed by a single individual. For example, simple tissue, complex tissue அப்படின்னு சொல்றோம் இல்லையா? The simple tissue அப்படின்னு எடுத்தா you are telling us parenchyma. It is a single individual. But whereas in complex tissue, it is formed by a group of components. For example, xylem means xylem, parenchyma, xylem, tracheids, all these will be grouping together to form a tissue. So it is a complex structure. Likewise, in articulated lattice also, it is formed by a compound of origin. Many cells will be grouped together to form this structure. Right. So that is known as compound lattice. For example, you can find the structure. So it is can also be represented as latex vessels. For example, Halia or Manihot. Non-articulated lattice, right? So in non-articulated lattice it is nothing but it is a very simple and it is formed by a single cell individual. It doesn't fuse with each other. But whereas articulated patina, those cells will be fused with each other to form a network-like structure. So that is known as articulated. So non-articulated is that they doesn't fuse with each other and it is a single cell in component we can find out, right? And the cytoplasm will be multinucleated. In a single cell, the cytoplasm will be more or less evenly distributed and the nucleus will be found more or less in the same place itself. Okay. So this is a latex cell. So it is formed of single component where you can find any nucleus and cytoplasmic content. Okay. So the thing that you have seen today is secretory structures. Right? Please have any clarification? So practically you have seen all these things, right? So whether you are able to capture the thing which I told you, it's okay. Thank you.